Good evening. Good evening to all. Today we are going to see about digital image fundamental. First, you have to know what is meant by digital image. It is we are representing digital image as a mathematical functions. The function is f of x comma y. Here f of x comma y is digital image. Here x comma y denotes the spatial coordinates and f represents the intensity of the gray level. So what are the process is going on in digital image? Here the digital image we are processing the all type of datas. That means we are processing the image, so we will get the digital image processing system. It covers low, mid and high level processing. In low level processing, input and output image are images. In mid level, the outputs are attributes and extracted from input images. In high level, an ensemble of recognition of individual objects. In digital image processing, we are representing each element as pixel. Pixel is the pixel representation is the picture elements. Pixel is nothing but picture element in digital image. In digital image, each pixel is represented with the spatial coordinate x comma y. That means x and y direction we are representing the digital image. Here we are processing the different types of stages in digital image processing. First one is that is we are acquiring the digital image, that means problem domain. It is what are the process we are going to adapt for that problem domain. First one is image acquisition. Image acquisition means how we are capturing the image. It is using the camera only we are capturing the image. Then from the camera the image is not in clear, so we have to improve the quality of the image. So that's why we are using the image enhancement technique. So image enhancement technique we are using filter concept. That means filtering. The image quality can be improved by two types of domain. One is frequency domain and another one is spatial domain. In spatial domain, here the process is carried on each pixel. But in frequency domain, that means we are representing the function in frequency domain. That is, we are taking Fourier transform of the function. After that, we are taking the inverse transform. We will get in spatial domain. Here, image restoration means we are reconstructing the image with the degraded image. That means we are recovering the original image from degraded images. Then we are going to done the morphological processing also, and we are segregating the small parts of process by using segmentations. From that, then we are recognizing the object and we are representing the image with different descriptions. And here the digital image size are very high, so we are going to compress the data. That is called image compression. And also we are converting gray level image to color image, so we are processing the color image data also. Here, this is the cross section of eye. Here we are using here we have retina, blind spot, fovea and lens system and corona, iris, all these there in the parts of the cross sections. Here the eye sphere is diameter is 20 mm. Here we have 3 membranes enclose the eye. Then we have two number of cells are there. There are two types of receptor in the retina. First one is the rod and cone. The rods are long, the cones are generally shorter and thicker. The rods and cones are not distributed evenly around the retina. Then the lens is colored by a slightly yellow pigmentations that increase with age. lens contains 60 to 70% water, about 6% fat and more protein than any other tissues in the eye. Here rods and cones operate differently. Okay, rods are more sensitive to light than cones. The number of rods is much larger, that means some 75 to 150 millions are distributed over the retina surface. The larger area of distributions and the fact that several rods are connected to a single nerve end and reduce the amount of detail discernible by these receptors. Next one, the cones in each eye number between 6 and 7 millions and they are located primarily in the central portion of the retina and is called fovea and are highly sensitive to colors. The cones are very highly sensitive to colors. So humans can resolve fine details with these cones largely because each one is connected its own nerve end. Muscles controlling the eye rotate the eyeball until the image of an object of interest falls of the fovea. Then cones responds to higher level of illuminations. The response is called photopic visions. What is meant by photopic visions? Cones responds to higher level of illuminations, so that is called photopic vision or bright light vision. The other name of photopic vision is called bright light vision. So cones responds to high level of illuminations. Here distribution of rods and cones are described in this curve. Here the absence of receptor in this area result is called blind spot. Here in graph we mark as blind spot. What is blind spot? Blind spot is the absence of receptors in this area. Then fovea itself is circular indentions in the retina of 1.5 mm in diameter. Except for this region, the distributions of a receptor is radial symmetric above the fovea. Receptor density is measured in degrees from the fovea, that is in degree of axis as measured by the angle formed by the visual axis and a line passing through the center of the lens intersecting the retina. Here the image size is shown. Here in ordinary photographic camera, the lens has a focal length and focusing at various distance is achieved by varying distance between the image and working plane. Here the focal length of the eye is 17 to 14 mm and h be the height in millimeter of the object and the retinal image is, the value of h is we can calculate 2.5 mm for this example. So the retinal image is reflected primarily in the area of the fovea. Here perception takes place by the relative excitations of light receptors which transform radiant energy into electrical impulse and denoted by brain. So the range of light intensity level to which human visual system can adapt on the order of 10 power of 10. Okay. The current sensitivity of the level of the visual system is called brightness level. What is meant by brightness adoption level mean? The current sensitivity level of visual system of our visual system is called the brightness adoption level. Photopic vision range is 10 power of 6. Okay. Here the graph shows the photopic visions and scotopic visions. It tells the difference of scotopic vision and scotopic visions. Here's why we have smaller Weber ratio indicates good discriminations of small percentage change in intensity discriminates. But in larger Weber ratio indicates poor discrimination large and percentage change in intensity required. Here the slide indicates the match band and simultaneous contrast. In match band effect, all gray level images can be represented. That means from white to dark we have different gray level values are explained. But in simultaneous contrast, that is we are placing the image with different background. Same intensity can be reflected or the same intensity of the image is placed in the different background gray values. Here it is image sampling and quantization is represented. Here image sampling means we are sampling the signal x and y. That is, we are taking one image, then we are partitioning the image AB. That is, we are taking one straight line AB. Then we sample the line AB. Then after that, quantization means we are digitizing the amplitude values. But in sampling means we digitizing the x and y values. So that is represented with the examples here. This is that means we know that quantization means rounding the values. So we are rounding the value of amplitude of our image. That means f, the value of f value is indicated here in digital. Then we are representing image by matrix form. So we are representing m with m cross n. So f of x comma y can be represented with the m cross n numerical array value. We can represent the digital image. A0 comma 0 represent the row wise. A0 comma 1 represents column wise images. That it is represented by f of 1 comma 1, f of 1 comma 2, dash 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 f1 comma n. In last row we are representing if we are taking m cross n pixels. Here discrete intensity interval. The number of bits required to store image. The number of bits required to store m cross n digital image means that is the value is m into n into k. So the different values of n and k it is placed here in the table. What is the bandwidth? So if we are increasing k value, we are using higher number of bit we are using. Then we will see about what is the basic relation between pixels. That means neighborhood, adjacency, then connectivity, paths, then region boundaries. Neighbor means a neighbor of pixel at coordinate x and y. 4 neighbor of p denoted by n suffix 4 of p. That means x minus 1 comma y, x plus 1 comma y, x comma y minus 1 and x comma y plus 1. Then next relation is 4 diagonal neighbors of p. It is denoted by n suffix d of p. That means d denotes diagonal neighbor. So x minus 1 comma y minus 1, x plus 1 comma y plus 1, x plus 1 comma y minus 1 and x minus 1 comma y plus 1. Then for 8 neighbors of pixel denoted by n8 of p. That means n8 of p, n4 of p and nd of p. Adjacency, let V be the set of the intensity value. Then 4 adjacency means 2 pixel and P and Q with the values from V are 4 adjacency if Q is in the set N4 of P. What is meant by 8 adjacency? That means 2 pixels P and Q with values from V are 8 adjacency if Q is the set N8 of P. That is represented here. Here path means a digital path or curve from pixel P with coordinate XO, YO to pixel Q with coordinate X and Y1 is a sequence of distinct pixel with coordinates. This is represented here. Thank you.